Alright, welcome back. Mr. P here. We are going to pick up where we left off. We left off talking about the prep step. Remember, this is the transition reaction or the link reaction. And remember, in the prep step, we took one molecule of pyruvic acid, and remember, 2 pyruvic acids are produced per glucose, but we're just going to show kind of the preparation of one. Notice this happens twice, okay, per glucose. But one molecule of pyruvic acid will be oxidized, meaning we're going to take 2 electrons and 1 hydrogen ion. We're going to give them to NAD+, reducing NAD+ to NADH. NADH is going to go to the electron transport chain, or the chemiosmosis process. And when we take these electrons and hydrogens off, it's going to remove this carbon, which will be released as carbon dioxide. Remember, this coenzyme A has to jump on to this 2-carbon acetyl, creating acetyl coenzyme A. And the coenzyme A's job is to act as that usher, and it's going to usher or lead this acetyl into the Krebs cycle. Okay. Krebs cycle is also called, I kind of messed this up. It should be the citric acid cycle, is also called Krebs. Okay, citric acid cycle. And so we have a Matrix movie meme, if you will. And so what if I told you the citric acid cycle happens in the matrix? Huh. Obviously, it's not this matrix, but it is the matrix, which is the fluid inside the mitochondria. Okay, so we are now leaving the cytoplasm. Remember, glycolysis happens in the cytoplasm. The Krebs cycle, which is the second process, happens in the matrix. So we have now we've transferred through the membranes, both the outer and inner membrane, and now we're inside the matrix where the Krebs cycle will take place. So, Krebs cycle, just like the Calvin cycle, is going to happen in a series of steps. This whole process takes place in the matrix, and we have to start and end with the same molecule, just like we did in the Calvin cycle. In this case, we're going to start and end with one molecule of oxaloacetic acid. Okay, one molecule of oxaloacetic acid. It is a 4-carbon molecule. You'll see these 4 little dots right here. That is a 4-carbon molecule called oxaloacetic acid. Yes, you need to know that name. One molecule of oxaloacetic acid is going to bind to the 2-carbon acetyl molecule once the coenzyme A drops it into the Krebs cycle right before the coenzyme A leaves. And so that coenzyme A is going to go back to the prep step where it's going to grab another acetyl and bring it back. Okay, that coenzyme A is kind of like a taxi that taxis the acetyls from the prep step into the Krebs. Okay, so a 4-carbon molecule is going to bind with a 2-carbon molecule. That's going to lead to a 6-carbon molecule. That 6-carbon molecule is called citric acid. Yes, you need to know that name. That's the reason why this molecule or this cycle is also called the citric acid cycle. Okay, because you are producing in step 1 citric acid. Oxaloacetic acid, 4 carbons, binds with a 2-carbon acetyl, which converts it to a 6-carbon citric acid cycle or citric acid. In step 2, we're going to take the 6-carbon citric acid. We're going to do the first of 2 oxidative decarboxylation events. Okay, oxidative decarboxylation, remember, is the removal of 1 carbon. We're going to cut that carbon off. We're going to remove it as CO2. It's going to leave the cycle, and it's going to go to the atmosphere. Okay, we release carbon dioxide to the atmosphere every time we breathe out. Okay, how do we release carbon dioxide? It's oxidative decarboxylation. We are going to cut a carbon off of this citric acid, and if you cut a carbon off, we're going to have a 5-carbon molecule. Okay, this 5-carbon molecule is alpha-ketoglutaric acid. That you don't need to know, but you can if you will. It's obviously best. When you remove this carbon, just like in the prep step, we have to oxidize this citric acid, meaning we're going to lose electrons. And so where are those electrons going to go? Those 2 electrons and hydrogen ion are going to bind with our NAD+, converting NAD plus into NADH. NADH is going to go to the electron transport chain. Okay, just like NADHs have always been going through Krebs and prep step and glycolysis. So we're going to oxidize this molecule by reducing NAD+. NAD+ is going to take 2 electrons and a hydrogen ion, converting it into NADH. When we do that, we remove a carbon dioxide and we are left with a 5-carbon molecule. That 5-carbon molecule is alpha-ketoglutaric acid. We're going to do the exact same thing again. We're going to oxidize this molecule. NAD+ is going to come in. It's going to take 2 electrons and a hydrogen ion. It's going to convert NAD+ into NADH. NADH, just like it did up here, is going to go to the ETC. We are going to cut a carbon off. So we're going to cut a carbon off of this 5-carbon molecule, resulting in a 4-carbon succinic acid molecule. Okay, so all we've done so far is we've taken a 4-carbon oxaloacetic acid, we've added 2 carbons, which is an acetyl, to it, creating a 6-carbon citric acid. Then we cut a carbon off, oxidize the molecule to reduce NAD+, NADH goes to the ETC, and we're left with 5 carbons. Then we cut a carbon off, we oxidize this molecule, we reduce NAD+, and we're left with a 4-carbon molecule. Now, you'll notice this step right here. This is another substrate level phosphorylation event. Okay, this is SLP. Now, SLP, we said, happens in the cytosol, or happens in the cytoplasm. It also happens in the matrix. It also happens in the stroma. Okay, so SLP, this is not the main way of producing ATP. We've talked about it before. This is the bonus ATP. We're going to produce 1 molecule of ATP per acetyl, or per cycle, and this cycle happens twice per glucose. Okay, why does it happen twice? Because you have 2 acetyl coenzyme A's produced per glucose. So we're going to produce 2 molecules of ATP just as bonus while we're doing the cycle. So we are left with a 4-carbon succinic acid molecule. Now all we're going to do in step 3 or step 4 is we're going to take this 4-carbon succinic acid. Notice we're not releasing any more carbons because we want to end and begin with a 4-carbon molecule. But we're going to take this 4-carbon molecule, we're going to squeeze it for a few more electrons and hydrogen. And so we take FAD, which is a new energy carrier, and FAD is going to take 2 electrons and 2 hydrogen ions this time. Okay? If you take 2 electrons and 2 hydrogen ions, then you end up with FADH2, and FADH2, just like NADHs, are going to go to the electron transport chain. Then you're left with malic acid. Okay, 4-carbon molecule malic acid. Malic acid is no different in the number of carbons. The only difference is it has 2 fewer hydrogens and 2 fewer electrons than succinic acid. Then we're going to squeeze malic acid for a few more electrons and a few more hydrogens, and all NAD+ is going to do is it's going to take 2 electrons, just like it always does, and a hydrogen ion, and it's going to reduce this molecule into NADH, which also is going to go to the electron transport chain. Notice this, 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 and this all are in yellow, and the purpose of those is to send them to the electron transport chain. What are we ultimately going to produce a lot of at the electron transport chain and chemiosmosis? We're going to produce tons of ATP. Okay. Is the purpose of the Krebs cycle to produce ATP? No, we only produce 2, okay, per glucose. That's not very much at all. The purpose of the Krebs is to produce high energy electron carriers, in that NADH times 6, because remember it happens twice, the cycle happens twice. And so if we produce 3 per cycle and it happens twice, then we are producing 6 total NADHs, and we're producing 2 total FADH2s. We're also producing 2 total ATPs. Okay, so we're going to start and end with the 4-carbon oxaloacetic acid. Substrate level phosphorylation, I'm not really going to go through. We talked about it in chapter 8, but just as a repeat, the slides are here. You can pause it, look at it. Remember, substrate level phosphorylation is when an enzyme takes a phosphate from an organic molecule, gives it to ADP, and produces ATP. This can happen in the cytoplasm or anywhere within the matrix or stroma. Okay, that's where we're going to end this one. See ya.